Hi, my name is Kevin and welcome to How to Tissue Culture. Today we're going to be focusing on hops, specifically stage 1 of tissue culture and how we're going to clean our material and put them into vessels. In this video, I'm going to be focusing on how to make a sterilization solution that we can use for our hop plants and the process of putting your cuttings that have been cleaned into fresh media in plant culture vessels. So join me, let's go to the lab and let's get to it.
All right, so right here, what you're gonna do is just make sure you clean out your workstation very well. Spray some ethanol. I was using a 70% solution. And then right here, I'm wiping it with the big Kim wipe. Make sure to get all the back area. You're really trying to have a clean starting space. It's gonna make everything a lot easier. All right, so here we have bleach. It is a 7.5% solution. We have some tween that we're going to use to create some soap suds, a plastic pipette, a glass test tube. It's optional. It's a 500 milliliter glass bottle, which is where we're going to put our solution. And we have some autoclaved water. And also a 50 ml graduated cylinder. All right, so now that we have all these things together, we're going to go ahead and pour our solution into our vessel and mix it in there. So first we're going to start with our bleach. So let's start and pour that in. We're going to use 240 mils, which is 80% of the 300 mils we're going to use for the full volume. And we're going to put the rest, the 60 mils of just the RO autoclaved water. All right, so now we're going to add some tween. For 300 mils, I added about 10 drops. Go ahead, drop that in. Try not to waste resources when possible. Tween is, it's just what makes it soapy. Helps the cleaning solution work a little better. And then you're gonna seal that up again. Still trying to keep a sterile approach, right? By like keeping the cap aimed away from you and give it a little shake and a little agitation, get the suds going. All right, so now we have a 91% ethanol, which is going to be the first part of our sterilization, and a bleach solution with tween. Yeah, thumbs up, baby. All right, so now that we got our cuttings, I'm going to go cut these from the greenhouse of our hop plants. We're going to go ahead and take these babies out. And we want to go ahead and kind of just get our cuttings to size. So right now, it doesn't really matter that we're not 100% being sterile, only because the plants themselves are not sterile right now. So I'm just going to go ahead and pull them out and use the table in my space to work with them. My hands, it's going to make everything a lot faster. You can honestly do this over the sink, but I'm just doing it right here in the hood because it's just, that's where all the tools are set up. Alright, so I'm going to go ahead and just take a few cuttings here and just trim off any excess that I don't want. So we're going to try to get the cuttings down to size, something that's manageable. And so here I'm going to cut ends, like the ends of the leaf, or where the leaf starts, and kind of leave a little bit of the stem portion. The stem is going to allow it to kind of protect itself when it's being sterilized, especially in the bleach solution. You're going to see how some cells are going to turn white, and that basically means that they're dead, but there's kind of like a layer of absorption, and not all of them die. It doesn't get like, it does take on a few centimeters of the plant, but not a lot. It's usually a very tiny amount, and it depends how strong your solution is. The stronger your bleach solution is, the more cells are kind of going to turn white, and you're going to see a little layer at the ends. All right, so I just emptied out the rest of the water, and now I'm going to put all these nice little cuttings into my glass container here. All right, so now that the cuttings are in the container, go ahead and wipe off your station again. Just to make sure we're going to start off with as clean as possible. Spray your hands. You know, it's like using hand sanitizer, but it's a little harsher because it's just 70% ethanol, so I wear gloves. I don't want to just spray my hands, but you know, you can technically do this without gloves. Alright, so here I'm just doing some water agitation. I'm gonna shake it in water for about 30 seconds. You could do it with your hand. You can also use a vortex machine. It's really up to you though, but I'm trying to make this as practical, as simple as possible. Hopefully I've been helpful, and you guys have been learning something, or at least just been entertained by my interesting science. Okay. Alright, so you're gonna go ahead and pour that out. And try to hold the cap so you don't lose any cuttings, because at this point you really don't want to, you don't want them falling into that water. It's all the dirty waste. Make sure to have a waste container, guys, if I haven't mentioned that already. Alright, so go ahead and start now. Pour your ethanol in, and now this is going to start the disinfecting and sterilization. Go ahead and shake that in there for another 30 seconds. Go down there, right there, getting cleaned up. Those poor little babies. And then you go ahead and pour that out. After you're done agitating it, shake it up and pour it out. Try to hold the cap kind of away from you, and try to hold any clean vessels away from you when you're doing this process. Alright, so now we're going to use our bleach solution, and we're going to add about 50 mils. We also used only 50 mils of the ethanol. So we're going to cap that up, and now we're going to shake them in there. Another 30 seconds. And after those 30 seconds, you pour it out. I'm editing out the shaking, and really, there's no point of keeping it in there. If you guys wondered that it didn't look like 30 seconds, that's probably why. But now after that bleach solution, go ahead and use some autoclave water, and use 50 mils again. We're going to do this rinse about 3 to 4 times, or as many times as you need to get all the suds out, which, you know, are pretty much left in there by the tween and some of the solution. But yeah, and at this point your cuttings are really clean, or as clean as they're probably going to get based on the concentration of your solution. So, and the amount of time they're in there, focus on trying to not lose any. Sometimes you will lose some. Sometimes they'll get stuck on the edges of the cap, or the edges of the bottle, and it's a little hard. You could tap it in, kind of like I tried there, but I believe for this time I lost about 2 cuttings. And it's okay. Like, it's not the end of the world. Just, you know, make a few and be careful. Here again, I cap it up, shake it up, and I've been rinsing this now for, I think 2 times. And here they are. Well, now that we're finally done. Alright, so now that they're nicely cleaned, you gotta spray your hands again and clean up your station. You really want to make sure you're clean at this point. Spray your hands up, clean them off, and here we're going to take out our petri dish, our glass petri dish to work on. One of those sides is going to have water in it, again autoclave water, and I stick my tools in them because I want them to cool off. They're in a heated sterilizer with glass beads. It's cranked up to 300 degrees Celsius, so they're very hot, but you know, dipping them into the water allows them to cool off quickly. So here I am taking the cuttings out. I poured them into the water just to keep them from desiccating. And so I'm taking some cuttings out. Next time I will put a paper towel or a Kim wipe under this, just to make sure that you guys can see well, because it's a little hard to see. I apologize. And I tried zooming in, but I kind of didn't make that much of a difference. Here we are though. And basically I'm trying to show you guys the white cells from the tips. They're basically the top end and the bottom end there that I'm pointing at right there. The top, you can see how it's a little white. It kind of died off because of the bleach. You know, they bleached the plant cells. And so those parts you're going to cut a little below them, so green tissue is left. You cut off all that white dead cells. And now you have them again in your petri dish, ready to be placed into plant vessels. And this is the exciting part. You're gonna put these cuttings straight into the agar. You're gonna ground with your freshly cleaned tools. And to clean your tools, you just take them back into the heat treatment. But here you go. So I'm basically just sticking in all the tips, all the shoots, right into the agar, into the media. And so this media contains half strength MS, sucrose, and no plant growth regulators. I'm not using any because I don't want to stimulate growth right now. I'm kind of just focusing on seeing if any of this tissue will not grow bacteria and stay clean, not grow any mold or from any spores. So hopefully we have clean tissue here, and this was a good example of how to do stage 1 with hops. I'll definitely make an update video on this process and on these plants here. There you go. Now what you want to do is make sure to label your vessels. And there you go. You have your tissue culture vessels. Good job, guys. There I am. I'm pretty proud.
All right, guys. Well, if you made it this far, let me just say I appreciate you, because this is my first YouTube video I'm publishing on this kind of work that I'm doing. I've been doing this plant tissue culture for about 1.5 years now. I started in grad school, and now I've graduated and I'm working out in the field, and I'm super happy that I get to do this kind of work because it's pretty great. I love doing plant tissue culture. It's exciting. It's neat because you get to play with plants and in the lab. So you can also go out to the greenhouse, right? When you take cuttings and go out to the field. So there's a lot of different methods and projects that you can work on and do at the same time. And I'm excited to make video content on a lot of it. So stay tuned. And if you like this kind of content, go ahead and subscribe. I'll see you guys soon.